Good morning, everyone. I'm glad to see that you braved the thunderstorms and showed up on time. I was actually a little worried that I would be late because the roads were completely a mess on the way in. So over the next 40 or 45 minutes, I just want to address a simple question: how to use metabolomics in your research. So this is mainly meant to be a background and sort of give you some sort of a feel of what the rest of the 2 days are going to be as well. And I will also show some examples of how metabolomics can be used. So the overview slide that you have, and it's all of this is in your handouts, so you don't have to pay more attention to these talk actually. So I will address what is metabolomics, and I'll address the role of metabolomics in systems biology, and briefly introduce the concepts of what the platforms for metabolomic analysis are. And show you some illustrative examples in biomedical research and using metabolomics and how it can be integrated. And some of this work is actually my own, and some of this work is going to be some of the recent papers that actually came out using microbiome that have generated a lot of interest and many press, science, including Science Friday, which my daughter actually came and told me that, this is something that was addressed in Science Friday by NPR, so it's an interesting thing. And finally, I'll stop with the limitations and pitfalls of this analysis. So the first question is, what is metabolomics? So here is one of the things that came about when the ARA funding was there, and which actually said this is a new pathway to discovery. And metabolomics technology development was considered to be one of the most novel and most innovative ways to actually understand systems biology. And they said this emerging field of metabolomics seeks to understand all small molecules found within cells or tissues. And I actually got my first R01 based on this RFA, which was actually a good thing for me personally. But then I went back and asked, is this truly an emerging field? Or is it something that has been there for a long time? And the answer is quite interesting, and it is really not. Is it not truly a new emerging field? In fact, as some of you may recognize the name, Linus Pauling here has done this many years ago. So he actually published a paper in 1971 in PNAS, and actually he looked at quantitative analysis of urine, vapor, and breath by gas-liquid partition chromatography. What is really interesting about this paper is that, those of you that are clinicians will remember that in the past, people used to be admitted forever in the hospital for 2 or 3 weeks. And what he did, he was interested in diet and antioxidants. So he used to admit people for 3 to 4 weeks in a GCRC setting, both normal people and people with disease, and basically give them various diets and try to understand how metabolism really worked. And of course, nobody gives us money to do these anymore. But this is telling that if you go back to this paper, he collected 24-hour urines in these patients, evaporated it, and actually looked at only the metabolite concentrated, and did a very fabulous analysis of more than about close to 200 metabolites. And in fact, we still do the same thing. They use the same gas-liquid chromatography. There's really not much that has changed, except that the tools that we have now are a little more automated and computer-generated. So what he called this field was called orthomolecular medicine. And I think this is the best definition of metabolomics I've come across. And I would use it all the way again. So what he said was orthomolecular diagnosis is the process of determining concentration of various substances in the body. Most of our experiments in orthomolecular diagnosis have involved use of gas-liquid chromatography, ion exchange chromatography, and mass spectrometry. And he just basically goes on to say in this paper he will discuss the methods. In fact, none of this has changed. We use exactly the same thing as you'll hear in the subsequent talks, and I will also illustrate this down the road. So the second question I want to address is, what is the role of metabolomics in systems biology? So a clinical phenotype of either a normal functioning individual or a disease or this could be an animal or a cell culture or whatever. It's a complex interaction between the environment and the composition. You know, you can have genetic, epigenetic, transcription, protein, metabolite, et cetera. So the role of metabolomics is as a part of the multi-omics platform for you to really understand at the multi-level analysis of the entire physical shape of a, you know, and the machinery of a cellular machinery of at homeostasis, which causes homeostasis. So this is a very simplistic view of how this whole paradigm works. I'm a nephrologist, so I have to have a kidney there. This is actually a cartoon put together by one of the clinical fellows who worked in my lab before. And I think it's, for the most part, it is kind of simplistic, but I think it sort of illustrates the point. First of all, all of these are not direct, you know, one-way directional thing, because there are a lot of interactions that actually can interact and change things. But overall, you know, you were talking about roughly about 25,000 genes in a human cell. As far as the transcript goes, if you look at alternate splice variants, the complexity markedly increases to roughly 100,000 transcripts or so. And if you look at only the primary outcomes of the transcript, the native proteins, that is about the same as the number of transcripts are. But the complexity of the protein part, the so-called proteomic analysis, is far more because of the fact that you have this post-translational modifications. So for example, you can have glycosylation, acetylation, phosphorylation, et cetera. So many of these post-translation modification can make the protein much more complex, more than 1 million or even 10 million as people think currently. Whereas the metabolomics is a far more defined. Again, the complexity narrows when you come down to the metabolite level. And again, I will illustrate why the total number, again, is a problematic, because it's actually dependent on the analytical tool that you use. The very databases currently think about between 3,000 to 20,000 or up to 30,000. And as you can see, this is the most proximate event to the eventual clinical phenotype. So this is something that, you know, obviously, if you can think about this as a sort of a final common pathway towards metabolism in terms of expression of the phenotype. So what are the advantages of metabolite analysis? So first thing is metabolites are readily obtainable in biofluids and freely filtered in urine and plasma. So you can actually measure this in people and animals and cells and whatnot. Now, we currently use metabolomics, as Chuck was briefly pointing out. We use, for accurate diagnosis of many diseases, we use metabolites. We use glucose for diabetes, cholesterol for hypercholesterolemia, and creatinine for renal failure. And metabolites are structurally unique and have a very well-defined nomenclature and a precise role in biological processes. So it's it's relatively unique in terms of pathway determination. They're stable compounds. In general, there's nothing called as post-translational modification of a metabolite. I mean, either it can be metabolized into a product or it actually stays on as it is. So it's actually relatively stable from that perspective. So in principle, metabolites represent the composite output of the cell. So it's accounting for the genetic, transcriptomic, and proteomic variability. So therefore, one could make an argument that this would be a better omic platform to study. Not better, but it's just a more reliably interpretable platform. So what constitutes a metabolome? So you can have metabolites in tissue and biofluids, but there are very many ways you can derive this. So first thing is obvious to everyone is metabolites can be synthesized by human cells or animal cells or the cell cultures that you have. More importantly, one should consider the diet. And as I pointed out, Linus Pauling's work was all done on diet, and he found marked variations in diet. And this is something that you really need to consider, especially when you're doing a clinical study, and also an animal study, and also a cell culture study in which you have the media. So you really need to figure out why, you know, what you've given contributes a lot to the metabolite pool. Metabolites can arise from commensal gut microbiota, and I will actually illustrate some studies that actually highlight this aspect. And metabolites can arise from pathogenic microorganisms, and drugs, including antibiotics, can be in the pool. And as antibiotics can also alter the microbiota, and that can have an impact on the metabolites that you see. Finally, environmental chemicals and toxins can also contribute to the metabolite pool and also alter the microbiota in ways that can actually alter metabolism. So you need to consider all of these things and the steady state between them when you try to interpret your data. So what about the plasma, and what are the metabolites that typically are there? So if you look at plasma and remove the proteins out of the plasma, about roughly 2/3 of the plasma metabolome are lipids, and about 1/3 are the rest. So you have nucleic acids, amino acids, and carbohydrates really constitute really 1/3 of the plasma. 2/3 are lipids. But importantly, lipids, this is by weight, by the way. So if you look at the lipids in general, they are higher molecular weight. And therefore, the number per se may not be as great. But clearly, the volume-wise, in terms of the lipids, are much more than what the rest of them are. In terms of lipids, you can actually have about 7 major classes of lipids in plasma. You have sterol-lipids, fatty acids, glycerolipids, glycerophospholipids, and sphingolipids, and a little bit of prenol-lipids as well. So if you look at the lipidome, it's one of the new platforms that we're going to be offering, and TM Rajendran here in the back is the one that is actually leading these efforts. And you will eventually have the expertise to really do these type of experiments in the core, and which is something that we will plan on offering. So I just wanted to throw this in and basically say that the lipids themselves are quite complex, and you have more than 400 to 500 lipid metabolites one can potentially measure down the road. And this is data that is derived from AB-SCIAX, which is an instrument that we are going to acquire in the core sometime during summer, and which potentially would be capable of doing these analyses. So going back to the databases, currently available databases, and asking the question, how many primary metabolites are there? This data is derived from Bishart's group in Alberta. So he notes that there are about 3,100 toxin environmental chemicals in this T3 database, and about 1,000 drug metabolites, and 1,450 primary drugs, and about 30,000 food additives and phytochemicals, and only 8,500 endogenous metabolites, what we call as endogenous metabolites, which are none of the above. So these endogenous metabolites are really the focus of many of our analysis, and not necessarily these things, but you have to understand that all of these actually do contribute to the endogenous metabolites as well. So what are the challenges of metabolomic analysis? So it's a large dynamic variation in terms of the concentrations. You have roughly 8 to 10 log-fold differences between the most abundant to the least abundant. So this is technically challenging just based on the fact that you have these large variations. As we talked about, diet and metabolic processes can alter these metabolites. And the chemical diversity of the compounds, the sheer nature of the compounds, makes it a daunting task. So if you look at the chemical structure, you have alcohols to acids to lipids. So really making comprehensive analysis of metabolome is quite difficult. In contrast to genomics, you have 4 base pairs. And in terms of proteins, there are mainly about 24 amino acids that can form proteins. So it's actually, it's a lot more analytically feasible for you to do those type of analysis as opposed to metabolomic analysis. So some of this will be addressed by Charles Evans when he gives his talk, but basically there are 3 major types of metabolomic experiments that one could potentially do. One is targeted analysis, which is mainly hypothesis-driven. So you can ask the question, what are the products of a specific enzyme, for example? And what happens to folate metabolism, for example? So you could basically pick up 5, 6 compounds and say, can I have a specific target analysis for this? These are much more reliable because you're really focusing on one group of compounds. We typically have a standard that we make our curve with, so it's actually very quantitative. And it's very, very reproducible. And in fact, many of the, for example, if you're using a drug screen for Lance Armstrong was convicted because of the fact that they did a targeted analysis of steroids by GCMS. And they could go back to plasma that was stored 20 years ago and come up with the thing that, yeah, you did use this drug. That was similar to native drug, but it was basically they could do it by a fragmentation pattern. They can actually determine it was artificially synthesized. Whereas untargeted, on the other hand, is we also call this unbiased or hypothesis generating type of an experiment, in which you just want to ask, qualitatively, what is different between the two systems that you're studying or three systems you're studying. This is by the nature of the metabolites that we are talking about, the diversity. This is less quantitative. This is only a qualitative assay. And less reproducible by the fact that we talked about. These are chemically diverse compounds, and you cannot have a single platform that really reliably, reproducibly measures all of these. And Charles will sort of take more on that part. And finally, the metabolic flux analysis, the fluxomics, is actually asking the question, how is one of these metabolites carried through the system? And typically, people use isotopically labeled substrates and look at their metabolism for this. So the next thing I want to address is analytical platforms for metabolomics. And you have all this fancy instrumentation that we have here right next door. And so the key things are the following. So the most important thing I would say is what goes on before the machine. The samples that you have are in a matrix, whether it's a cell culture, tissue, or a biofluid, which needs to be extracted out first and separated so that then before it is actually being detected with identification in some sort of a system. So these two steps are actually very critical, and Lee and Anna are going to discuss this further down the road. In fact, I actually think this is probably more important than the actual machine part, because we can reproduce the machine part much better, but this is going to be a very critical aspect of how you handle your data. So the two major techniques that people use currently, so NMR and mass spectrometry. So NMR, nuclear magnetic resonance imaging, it's not imaging, it sort of can be used as imaging as well, but you can detect compounds as well. So main advantage is there's no chemical extraction or derivatization that is required. The disadvantages are that it's lower sensitivity and limited number of analytes. Dr. Rams this afternoon will actually discuss a lot of this in detail. We don't specifically offer this as a core, but we can refer you to people that have NMR facility in the university for more work if this is what you need for your experiments. We mainly use mass spectrometry in our core. The major advantages are the high sensitivity, high resolution, and you can couple either a gas chromatography or liquid chromatography as a front end that allows you to really diversify your portfolio in a way in terms of your metabolic chemical diversity of compounds that you get. And the disadvantages are that there's a need for chemical extraction, and in case of GCMS, you need to derivatize the samples, which just makes it a little more cumbersome and complicated. So a typical workflow is something like this. I don't want you to memorize this, but basically, what we do is after you separate out your metabolite mixture, then it goes to some sort of a mass spectrometric analysis. Either you can have the untargeted or the targeted analysis. And we have a positive and negative modes of analysis. And then you get data. When you get the data, this is unfiltered data, which can be quite user unfriendly because you have to identify the various peaks that come out. The peak have to be deconvoluted and then aligned and filtered. And then we have to search the compound from a database and eventually sort of identify the compounds. For a targeted analysis, it's a lot easier because we typically have a standard and we can actually go back and do this much more robust way. For untargeted, this actually involves a lot of work. And Sasha was actually going to be doing a sort of a session tomorrow, actually going through how we do this for untargeted analysis. So once you have your analysis, you have this normalized entity list. You normalize using various criteria that you develop, and eventually you can do some statistical analysis and then do some confirmation or validation experiments using more targeted type of an analysis. So this is a typical workflow. So I'm just going to go for some illustrative examples and show you. So this is one of the first studies that we were involved in. TM and Jamin in the back, well, both of them were involved in the study as well. This was done with when Arun Sri Kumar was here with Arul Chinayan's group. And so this is your dreaded heat map that you have. As you know, we have also sort of learned from our genomics colleagues and used this for expressing these type of data sets. Now, this is a study done in prostate cancer. And there are three different groups of patients. You know, you have benign lesions, localized prostate cancer, and metastatic prostate cancer. And so you can obviously see from the thing that there are some differences in colors you can see, although the actual granularity of the data is not there. Now, one way of expressing these data and doing this is actually asking the question, how these two, localized cancer and metastatic cancer, are different from benign. And for that, you actually do a standard thing, saying basically that, you know, you have basically a bell-shaped curve, and you ask the question whether it's how many standard deviations it's something is different from the mean. And you can see that if you look at the deviation from the benign, you can see that localized prostate cancer, so there's a lot of different things that are different. And in metastatic disease, there's a big differences in many different pools that you come up with. So here's what the metabolomic signature of a clinically localized prostate cancer was. And there was a bunch of different things here, and these are ranked based on the deviations from the benign. And you can have 5, 10, 15-fold standard deviations apart, so you know that it's highly statistically significant, most likely. And what turned out was that you have an elevation of amino acid pools in localized cancers, essentially. And there are a bunch of different amino acids that were actually different. So then the next question came about is, can we pick up one of the stronger signals from this? And as I told you, this is a qualitative analysis. So just because it's 5 standard deviations apart does not make it it's 5 or 10-fold increase or whatnot. It's just a qualitative difference. So you need, in order for you to really understand what the concentration is, you have to develop a targeted analysis focusing on the quantitative information. So that was what was done. Jamin and TM actually developed this assay looking at sarcosine, one of the markers that was actually done. They did a GCMS analysis for this, and what you find is quite remarkable. So you have this is femtomoles. This is very quantitative, targeted analysis. The benign versus local prostate cancer and the metastatic disease, there's a huge difference in this molecule, and this is represented in sort of a box whisker plot here. And this turned out actually to be validated in 3 large trials, and recently I just saw that recent paper that actually a very large European trial actually validated this as well. So this was the first study, and essentially it's been validated in other studies as well. Now the classic, I want to go to the microbiome. There's a lot of interest in this and metabolism of microbiome, and our own core has been doing a lot of work related to the microbiome. So I just wanted to highlight some of the recent studies that have come out that are quite interesting. So this is sort of the first study that really came about by Jeff Gordon's group, when they actually found out obesity-associated gut microbiome that actually increased the capacity for energy harvest. So what they did was in this thing was quite simple. They looked at a lean mouse and an obese mouse, and they looked at the cecal contents for short-chain fatty acids. Remember, the short-chain fatty acids are usually generated by bacteria. And essentially, they're looking at levels of the short-chain fatty acids. This is acetate, propionate, and butyrate. What they were able to find is that obese mice had a higher level in the gut of these short-chain fatty acids. So what this actually implies is that there's some sort of alteration in the microbiome that is actually generating these. And they then did a bomb calorimetry of feeding these mice the same diet of the stool to determine how much was the energy extraction from the stool. And remarkably, what they find is the obese mice actually extract a lot more of the energy from the food rather than rather than the lean mice. So that is the. So this is the lean mice. This is the obese mice. Obese extract a lot more. So then the final interesting aspect was that what they did was they had donors, which are obese mice. They took the transplant of the cecal microbes and put it in a germ-free lean mice, and actually measured total fat composition. I believe it's 3 to 4 weeks later. And what they find is that the increase in body fat was fairly significant, in only in the donors that were actually transplanted by the obese mouse microbiome, suggesting that the microbiome actually can alter the amount of energy that you can actually extract out of your diet. But this did not answer the question whether diet actually alters the microbiome itself, which is the focus of the subsequent studies and some of the recent papers that actually came out of Stan Hazen's lab in Cleveland Clinic. And he discussed some of these when he was actually here a few months ago as well. So in this trial, Stan's group looked at what are the major metabolomic components that he can actually measure out of diet, out of defining the cardiovascular phenotype. So they had a learning cohort and a validation cohort, untargeted metabolomic analysis, eventually ended up with about 18 analytes that actually highly correlated with the clinical phenotype of cardiovascular disease, and 2 different major group of cardiovascular disease patients. Now, one of the neat, cool things they did was they actually looked at what are the major correlations and tried to understand whether the analytes were derived from the same pathway. And that is quite an interesting way of actually dealing with this untargeted analysis. And they came up with this interesting pathway called the choline betaine TMAO pathway. So one of the compounds, TMAO and betaine, actually were markedly elevated in CVD patients. So what they did was they found out that choline, dietary choline, could actually be the precursor for this compound. So here you have choline. If you take it orally, gut flora can convert choline to TMA, which is trimethylamine, which is actually a gas, which is the one that actually gives the bad smell to the colonic contents. Let me put it that way. So if this is absorbed, actually, then it's, the liver converts this in flavin monoamine oxidase into TMAO, this compound, which is what was elevated in patients with cardiovascular disease. So if you give choline intraperitoneally, the only compound it forms is betaine. That is what is actually mentioned here. So this is an animal model in which they gave this labeled phosphatidylcholine IP. And what you find is that both choline and betaine are elevated, whereas TMAO is not. Whereas if they give it orally, you have both choline, TMAO, and betaine are elevated. So basically, it needs the gut microbiome to form this compound. So they suppress the microflora and show post-antibiotics, you don't form any TMAO. And when they conventionalize this mice, reintroduce the microbiome, you form TMAO one more time. So basically indicating that this is a very specific compound for the gut microbiome. So what is interesting is they actually took it to the human studies. That's actually what is more remarkable. So they actually did, the question was the TMAO from dietary phosphatidylcholine, does this cause, does it actually predict major adverse risk factors for cardiovascular events? So, and this was the recent paper that was published, essentially showing that the TMAO, the top quartile compared to the bottom quartile, predicted myocardial infarction, stroke, or death in an incident population over 4 years. And this is baseline levels. They're following these patients for 4 years. It's a fairly large number of patients. You're almost 1,000 patients. Now, they were doing TMAO by quantitative mass spectrometry, a targeted analysis using standard. So this is very accurate and reproducible method. And this is the adjusted and unadjusted odds ratio. And you have a fairly remarkable increase in pretty much every single cardiovascular endpoint I looked at. So the idea here is that you have dietary phosphatidylcholine, and that is basically converted by the gut flora to trimethylamine, which is then converted to TMAO, then which actually increases atherosclerosis, potentially. So that's pretty much mentioned in the slide. Now, the other thing that they actually looked at was carnitine, which can also, is very chemically related to choline, and that can also form the same compound, essentially. So carnitine is actually used for mitochondrial fatty acid oxidation, and we have studied this, and many people have studied this in great detail. But it's also a major constituent of red meat, and a lot of energy drinks as well. So those of you that drink energy drinks, be careful of what you're putting in your belly, because carnitine obviously is a major source of is present at very high levels in many of the energy drinks. So what they detected was basically the same thing as they did for TMAO. Essentially, carnitine levels predicted incident coronary artery disease and peripheral vascular disease and CVD events. And what was interesting is that carnitine level, effect of carnitine was due to the TMAO. It was not due to, so if you have high carnitine level and low TMAO level, that did not increase the risk for atherosclerosis or cardiovascular events. If you had high levels of carnitine and a high level of TMAO, then you had the maximum events. So the carnitine was actually acting through the TMAO pathway, very similar to the way phosphatidylcholine was. So here's their summary slide from this paper, essentially showing carnitine and phosphatidylcholine. Both of them are altered by the gut flora into TMAO, which actually increases atherosclerosis risk. So there's so you can do many types of these type of analysis and actually try to figure out what would be a more appropriate thing for your own work. So I want to switch gears again and go to data analysis issues, the last 10 minutes I have. So one of the major questions that comes about is that how do you achieve greater metabolome coverage and known versus unknown metabolites, and whether how good is relative quantitation? What is the experimental reproducibility of these things? And the deconvolution and feature selection strategy. So these are the things that you will be addressed in more detail by others, but I just want to give a general overview of what is coming. So, here is a sort of a spectrum that you get. This is one of the analysis that we had done, but basically this is something typical spectra you get from a mass spec. So mass spec, you have a retention time in which you'll see the chromatogram in which your metabolite actually eluted off the column. And then you have a mass here. So each one of these points we call this as a molecular feature, which essentially identifies the compound by the retention time and a specific mass. Now you have this mess here, which is you have 3,742 compounds in an untargeted analysis. This is a simple urine analysis by electrospray positive mode. And then if you change the modes, then you get more or less, and some of them are linked and some of them are not. So this is really an analytical mess. And it does not necessarily mean each one of those metabolites are unique metabolites. It could be a sodium adduct, or you may have a loss of a proton, things like that. So, but you have to basically have an analytical strategy to really extract out meaning out of this data. And this was an experiment that was done by the Wellcome Trust group in the UK, which actually they show that how potentially irreproducible many of these untargeted analysis could be. Now, what they have here is basically a QC sample, which is basically a pooled plasma sample in this case. And they're showing data just for one metabolite that is analyzed. And these are the rest of the samples in the run. And you get varying peak areas for each of these metabolites. But when they ran the pool in between across the board, for this particular metabolite, the pool actually starts here. The same pool over time actually is quite varied. So here, if you measure this compound here, this would be 4.5 or something. But if you measure it over here, this would be 2.8 or something. There's a significant variation associated with this. This is one of the reasons why we do QC and pools that actually Lee will address in his talk. But what you could do is, if you ran these QC samples, you could actually normalize this data and kind of correct these for the pool, and therefore you get a normalized value of your metabolite. And this is particularly important for untargeted type analysis. So what we normally do is raw data is processed, and then we say we have some sort of a QC thing for untargeted. We say at least it should be present in half the samples of at least one group. And then this is what the signal correction is, essentially correct for your pool. And then you have some QA, QC data about how much of standard deviation that we accept as potentially acceptable. And then you build your reference peaks, and then we actually come up with the final data set for further processing and analysis. And you could do this for GCMS, and you could do it for positive electrospray and negative electrospray, and combine all your data to come up with your final data analysis. And this is just for serum or protein. So the last part of the talk is going to be on metabolite databases. So the databases, I would say, that are fairly incomplete. And this is basically based on the fact that people have whatever is published in the literature is what the database is all about. So you have This is the human metabolome database. This is probably one of the highest cited metabolite databases that people actually work with. Was done by Matt Bichart's group. And currently, and they constantly update it. But you could see some of the problems with going just by the mass or even the retention time in your untargeted analysis. For example, you have an adduct with a molecular weight of 193.0739. This is very, very accurate, you would think, of a molecular weight-wise. But there are about 6 compounds in the database that has the same mass. And many of them are isomers, so they may not even have, they will probably have the same retention time on the column. It's impossible to really separate isomers off the column. So unless you have a real standard way of standards to actually come back and do this analysis, this can be difficult to deal with. A lot of times, people have actually introduced an additional MS called MS. Like you fragment this piece one more time and see if there's a fingerprint is different, and that's one way of doing it. And we are actually developing our own libraries used on MS, which actually Lee is leading those efforts in our core at this point. And there are many metabolomic databases and tools that you could use in the literature, the KEGG, the metabolic enrichment analysis, MetPA, et cetera, et cetera. Our own internal thing is Alla Koronowski has done a fantastic job in actually setting up this particular thing that we use. Is basically a Cytoscape plugin called Medscape. And what you could do is basically you could put in input gene expression data into metabolomics data, combine all of them, and actually get this compound network, compound reaction network, et cetera. And Alla is going to be doing a session, giving a talk, and also doing a session later to go through the nitty-gritty details how you could potentially use some of these tools in your own analysis. But in general, you have something like this. You have, you can input your compounds here and essentially look at these things into sort of a more visually pleasing way for you to interpret the data into publishable format. So you can actually look at these things and actually put it as a part of your publication. So I'll actually address the limitations of metabolomics, some of which I addressed throughout the talk. So the experimental conditions, sample prep has to be very, very clearly defined. And we will be saying this over and over for the rest of the time in the seminar, in the symposium, because I think this probably is the single most critical piece. And that actually happens in your labs. It doesn't happen with us for the most part. And so you have to be very, very clear about what you're looking at, because even minor alterations can result in significant changes in your metabolites. So no single analytical technique offers a comprehensive analysis of the metabolome. So each technique biases your analysis to one way or the other. So there's nothing called as truly unbiased metabolomics. That's why we use the term untargeted now. But it's still, you are going to enrich certain factors based on the analytical tool that you chose. The metabolite libraries currently available are incomplete and rather limited. And it's important for you to understand this. There's a very high risk of false positive data, especially with untargeted analysis. So for higher specificity, typically I recommend using 2 orthogonal properties. I mean, for example, a retention time is just a physical property of the compound on the column. And accurate mass or fragmentation like MS may be more important to incorporate into your analysis. So the reproducibility of the chromatographic separation is a problem, especially for LC. So we always use these internal standards and retention time monitoring, and instrumental variability is a problem. As I showed you data that the pool samples have different. It's very important for you to use QC samples for that reason. And the current deconvolution feature selection software is suboptimal as well. So this is very vendor dependent. So we are actually developing our own tools that are vendor independent to analyze some of these things. And the gold standard is always a targeted metabolite identification by doing sort of a typically what we call as MRM type analysis. And this way, you know exactly, you can validate your data that you derive from the untargeted analysis. So these are the folks that actually do the work. We just come up here and give the talk. And the folks that actually in the core that are very helpful to you, and it will be potentially can help with your analysis up front. And the sooner you talk to them, the better things are, because I think we can help you design your experiments. So I'll just briefly, what Chuck said, I'll just briefly go through the overview of our core. system. We have an analytical core, which is basically all the instrumentation. We have very different types of analysis we offer. The data core is where the spectra is derived and meaning is brought into the spectra. So this is cleaning the data, normalizing, et cetera. And the statistics and bioinformatics core can help you with the statistical aspect of things. And obviously, the promotion and outreach core is the reason why we are here today. They organize the symposium and doing the other, and they give you some pilot and feasibility grants if you're interested in actually doing some pilot experiments for this type of work. The core structure, Steve Brown is there in the back, right there, and he'll be giving a talk later. Contact Steve with any issues or consultation up front. That will be very helpful. Lipomics Lab is run by Arun Das, who's right in the middle here. Targeted Metabolomics Lab is run by Chunhai. I don't see Chunhai here. And untargeted by Lee. And essentially, the data and the LIMS infrastructure is done by Anu Jenga, and data integration statistics by Sasha, who will be giving a talk later this afternoon, as well as Tanu Soni, who I think has done a phenomenally good job in actually making this data more palatable to the investigator. Because this is complicated, complex set of data sets. So the rest of the day is going to be arranged like this. So I just want to briefly introduce the rest of the talks that come along the way. When you get an idea for a thing, any experiment, talk to Steve so he can help you with the design of the experiment. For example, the diet and things like that. You have to be very careful about how you plan your experiment. The numbers that you need, for example. And then Anna Matthew is going to address the study design and sample collection in the next talk. Li Zhang will talk about the sample preparation issues and what you need to think about in terms of QC and other things. The machines and techniques are going to be addressed by Charles Evans, who's going to talk about the mass spectrometry component of this, and Rams, who's going to talk about the NMR component of this. And data extraction of the initial step is going to be Tanu Soni, who just walked in. And the statistical analysis is going to be addressed by Kirby Shedden. And Alla Kornowski will talk about the informatics Medscape part. Sasha will talk about how the untargeted analysis is actually going to be analyzed. And finally, Chuck will end with case studies of common problems and pitfalls and actually go through some of these issues that I briefly address in my long overview. And I think I'm on time. It's 9:15. Thank you. Sure. Boy, did you get to sleep then? Yeah.
Showing call is wonderful, but I'm not 100% clear how you define a metabolite. What is it? Is it sodium and chloride metabolites too? Or
No. So when we actually do the deconvolution for the untargeted, we take into account the common adducts that come through. So for example, if you are an acid, the sodium metabolite is probably the most common one, right? So because you're going to form a salt. Those are, you can actually, based on the buffer you're using, we can actually use a specific set of data integration tools that would remove those items. So for example, if you have a mass of 300 and then you have a sodium addict, a particular sodium addict would be added to that and would be subtracted. And you'll get, or actually would be added.
I'm talking more basic than that.
Yeah.
I think we're talking about organic molecules.
Organic molecules. We are just talking about organic molecules.
Okay.
Yes.
So amino acids are, where do they fall?
Amino acids are metabolites.
So that you could call that, but once they're put together into proteins, we don't call them metabolites.
That's correct.
Anymore.
That's correct.
In general, they're thought to be unpolymerized small organic molecules.
Okay.
Right.
Polymerized acetylcholine is our fatty acids, but we have to parse it somehow.
Right.
But
So
they're also
typically,
independently functioning
the entities.
entities.
Right. And the other thing, you could make a size and criteria. You can say less than 1,000 kilodaltons. A lot of people do that part. But again, some of the polypeptides do come in these things. It's not entirely functionally. Many of our unknowns are actually polypeptides.
Right. So, I was thinking back as your definition, you put it by Linus Pauling, and that was pretty broad
Right.
in terms of what he's looking for.
Right. Yes.
The effect was really great, but if you have, for your Pennathur or versus from other universities, from another university,
Yes.
how does this interaction? How they envision it?
So you would start with Steve, actually. So that would not change. Yes, by phone, and will have the information for you, and when he gives his talk as well, and it's probably in your handout somewhere in the back. Now and you would start with Steve, and essentially that would be your conduit towards what type of consultation you need. Some people need a very small amount of, you may already have an assay up and running, and you may have a very limited interaction with the core, or you may need extensive interaction, and we are here to help. Whatever be your need, we'll be more than happy to work with you. All right, so thank you. And I have a list of references after the slide. So you have all the references I talked about are already there in your handout. So you don't have to worry about any of these things. So basically you have, 14 main references. So you can go back and look at those.